Hello, my name is Mike Ward. I'm the Head of Content at Informa Pharma Intelligence. We're here in Vienna at the BioEurope Spring Meeting, where pharmaceutical companies, biotech companies, investors all get together to sort of discuss the trends that are dominating the industry at the moment, but also looking for opportunities for partnerships and investments. One of the hot topics of recent years has been the rise of advanced therapies, cell therapies, gene therapies. This is something that the potential has been known for a long time, but in recent years we've actually seen that come to fruition. I'm joined by Nicole Faust, who's the CEO of CEVEC. Manufacturing of cell therapies has always been one of the big challenges. What has happened in recent years that where it's developed?
Yeah, sure, thanks. That's an important question, of course. With the successes we're seeing in gene therapies recently, the manufacturing issues really need to be solved. And it's not only in cell therapy, it's also in gene therapy. And there, one of the big challenges still is that the technologies for manufacturing the viral vectors, which are basically the vehicles you need to transfer the genetic information into the patient for gene therapy, these methods, they have usually come out of the universities that have developed the therapeutic. So they are well suited for producing small batches, for early clinical studies, but when you move now to indications where you will need big production batches of such vectors, like for example, there are trials going on for Alzheimer using viral vectors, you really need to be able to have scalable production technologies, and that's not what the current systems that have come out of the universities can do.
Yeah, and what are the problems with those in terms of struggling with scalability? I mean, what is the usual challenge?
Yes, so to produce a gene therapy vector, which is a modified virus particle, so let me give you an example. AAV, for example, adeno-associated virus, that is one of the very safe viral vectors. It's naturally occurring in humans, it's non-pathogenic, it's quite robust, so it's the preferred vehicle right now. To actually make such a vector, you need a biologic production system, so you need cell lines to produce this virus. You cannot assemble them just in the tube. And these cell lines very often will grow on substrates. That means you have to provide a surface for the cells to grow on. That already tells you this is hard to scale up. And also you have to bring in the genetic information encoding for the virus in every single production run, either by so-called transient transfection, where you have to add plasmids, GMP quality. The gene therapy companies we're talking to are telling me the costs are prohibitive for these plasmids, or you have to bring in another virus. So all this makes the process expensive, hard to reproduce, and very hard to scale. And this is obviously where a solution is needed and where we think we are able to provide one.
So, Steve, you've developed a platform that is based around AAV.
Yes, we have developed a platform that helps producing AAV, and as opposed to the method that I just described, our platform also uses a cell line, obviously because that's needed, but that cell grows in suspension. That means it does not need the substrate anymore for tearing, so you can grow it up in a standard bioreactor, like you use it, for example, for antibody production, a method that's well established these days. And the second important step is we have modified the cells in such a way that they already stably carry all the genetic AAV vector information that's required. So we don't have, for a production run, we don't have to bring in any more DNA. So all we do is we grow up the cells and then induce the AAV production. So it's as easy as antibody production now with that system.
And with the regulators, because I mean this is still sort of a fairly new approach. Are the regulations in place that actually it's easy now to actually have a sort of scalable opportunity?
And so with the regulation for the cell line itself, that has been used for producing different types of gene therapy vector, adenoviral vectors, and these have gone to the clinic, so they have passed the FDA IND filing, which is always one of the strictest gateways that you might have to pass. And for the other methods that are like the stable integration and the cell line development and everything it takes, these are well-established methods coming out of the antibody production, recombinant protein production. So there's nothing particularly new to it. So we are not expecting any hurdles there.
Okay, great. So, I mean, this is sort of still fairly new. So, do you still have some examples of, what sort of proof of principle or proof of concept you've already, been able to achieve?
Yeah, so what we have done is we have generated such cell lines, and we have shown they are able to make the viral particles and also expressing a functional gene. I think this is something that is really important, and this has led quite a number of biotech companies active in the gene therapy field, but also the bigger pharma companies that are active in this area to start working with us to now transfer this technology to their specific products.
So how does that interaction work? I mean, do I come with you and say, look, I have this gene, can you help me sort of make a gene product? How's it structured?
Yes, so very often our clients already have something in phase 1, and then they realize, oh, this looks successful, it's great, but how am I going to produce for phase 3 or later on for market? I need a better platform. So they already know what the final vector should look like, and they ask us to transfer basically the production process then to our platform. So we know what the final product should look like, and we make the producer cell line for doing that.
Right, and the relationships that you construct, how does that work? I mean, what sort of, what would a deal look like if I was involved with, getting involved with CEVEC?
Yeah, so it's quite straightforward. So we start off normally by a service project. So it's a fee for service. We generate the producer cell, and if that works fine and it goes into production, then we we give out the cells under a license model, basically allowing our clients then to do the production with their CMO, or we can also arrange for the production with the CMO, and then there is a license for using the cell lines for production.
Okay, so that they would get a license to the technology, and that could actually then be embedded in a third-party CMO.
Yes.
And you would do. What sort of support do you give that CMO? I mean, do you still have to sort of give ongoing support?
We do the cell line development and the process development. We make sure it's a fully GMP compatible process, and we do the tech transfer to the CMO. And in the ongoing projects, we feel we initially still give them quite some support, but that obviously then gets less and less, and with the increasing number of projects, also you need to give less support.
Right.
But that's included in the deal.
So at the moment you're at a meeting like this, you're looking to attract new clients.
Partially, yes. So we're keeping in touch with our clients because it's good to see each other face to face once or twice a year. And we're also talking to new companies who might not have heard of us or not aware of the technology. And we've had quite some interesting meetings and discussions here.
Right. And you're based in Germany?
Yes, we're based in Germany, in Cologne, which is basically the center of Germany if you go north-south.
Right, okay. And in terms of sort of your development, are you looking to sort of expand beyond sort of the AAV space?
We are definitely looking into the other viral vector types, so lentiviral vectors is another important vector, for example, for the CAR-T therapies, which have become very famous. You will need a lentiviral vector, or for other ex vivo therapies. That is something where we have already seen in some proof of concept projects that the platform also is quite suitable, and that will be one of the next steps in our development activities to expand this to lentivirus. We are already quite active in adenoviral vector production because this is what the cells were originally made for many years back, and they have much better safety features than 2 and 3 cells for adenoviral vector production. So that's also something where we already have projects in, or our clients have projects in the clinic.
Right. And in terms of sort of financial support for the company, because I mean, obviously that kind of development still takes research dollars. Do you have enough financial runway to be able to achieve those ambitions?
Yes, we do. So we have enough by our current and also some new investors, enough financial runway now to really also make the last steps to industrialize the platform quickly, and then we're also generating increasing revenues and income from the service projects and from the licenses.
Right. Okay, well, Nicole, thanks very much for stopping by.
Thanks a lot, Mike.
Cheers.